Today, we're going deep, really deep, into the microscopic architect that built the very foundation of your body. We're not looking at genes or cells in general. We're looking at the uh, fundamental segmented blueprint that dictated where every single rib, every vertebra,
And every major muscle group would form.
exactly, in your core, your limbs, all of it.
This is the story of somites. And really, if you think about the basic organization of a spine and the muscles attached to it, that repeating pattern is, well, it's ancient. And it all traces back to these little structures.
So they're the essential building blocks.
They are. They transform a simple embryo into a complex, segmented creature.
Right. And our mission today is to take what is usually some very dense developmental biology and just boil it down.
Distill it, yeah.
We're going to pull out three things: the unbelievably precise timetable these segments follow, the three distinct destinies or fates of the cells inside them, and the molecular conductors,
The chemical commands.
the commands that orchestrate this whole construction project.
So just to start with a definition, a somite is basically an epithelial ball of cells. They form in symmetrical segmental pairs right next to the developing neural tube.
And they come from where?
They're derived entirely from the paraxial mesoderm, one of the early foundational tissues. The sources put it best. They're the segmented blueprint for your entire axial body structure.
Okay, let's unpack that origin story then. We have to trace the lineage all the way back to the epiblast.
Right, the source tissue for all three germ layers: the ectoderm, endoderm, and mesoderm. The paraxial mesoderm is that innermost layer of mesoderm sort of snuggled right against the midline.
But they don't just appear fully formed, do they?
Not at all. Up in the cranial region, the head end, the mesoderm, is first organized into these loosely structured collections of cells. They're called somatomeres.
Somatomeres, okay.
It's only when you move down from the occipital region, the base of the skull, and go caudally, that these segments condense and organize into the definitive compact somites that shape our anatomy.
This organization process is what fascinated me the most in the sources. The sheer precision of the timing.
Oh, it's incredible.
It's not some slow, creeping transformation. It's like clockwork.
It truly is a remarkable biological schedule. Somite formation kicks off during the third week of embryonic life. The very first pair appears
When?
in the occipital region around the 20th day of development.
20 days in, and the segmentation just begins.
And from that point on, they appear with astonishing regularity. It's a craniocaudal sequence, top to bottom, at a rate of about 3 pairs per day.
3 pairs per day. That's a crucial number.
It really is. I mean, if you think about the complex choreography of cell migration and adhesion required to form just one pair, maintaining that pace for weeks is just phenomenal.
So by the end of the 5th week,
The segmentation phase is complete, and you have a final count of 42 to 44 pairs.
42 to 44. And that breakdown mirrors our final body plan.
Pretty much. You get 4 occipital, 8 cervical for the neck, 12 thoracic, 5 lumbar, 5 sacral, and then 8 to 10 coccygeal pairs for the tailbone.
And here's a really interesting detail. Not all of them stick around.
No, they don't, and that's a key insight. The first occipital pair and the last, say, 5 to 7 coccygeal pairs actually disappear. They degenerate later on.
Which raises a question for me about efficiency. If this process is so precise, why build up to a dozen structures just to get rid of them?
That's a great question. It suggests that the underlying genetic program prioritizes a fast sequential segmentation first. It might be an older developmental blueprint,
So lay down the tracks first, then decide which stations to keep.
Exactly. The program is linear, even if the final product needs fewer segments.
Well, regardless, this precision gives us a fantastic tool. Because they appear with such a specified periodicity, that 3 pairs a day rate, the sources highlight that the age of an embryo can be accurately determined just by counting the somites.
It turns these segmented blocks into verifiable biological tick marks. It gives scientists and clinicians a highly reliable metric for dating early development.
Okay, so from counting them, let's move to their destiny. By the start of the 4th week, the somite differentiates. It starts as a simple epithelial donut of cells.
But then the cell fates diverge into 3 major components.
Right, the cells have to fundamentally change.
They do.
Let's start with the skeleton, the part that gives us our structure. The sclerotome.
The sclerotome forms from the ventromedial part of the somite. That's the lower inner part closest to the notochord.
And the key step is a transformation.
A critical one. The cells lose their tight, fixed epithelial characteristics and become mesenchymal cells.
Mesenchymal meaning what, exactly? If epithelial cells are like tight bricks in a wall, what are mesenchymal cells?
Think of them like wet sand or loose gravel. They're highly migratory individual cells that can move independently.
So they break free?
They break free and they migrate inward to surround both the developing neural tube and the notochord.
And what's their primary job once they get there?
They form the foundation of our vertebral column and the ribs. And not just that, the sclerotome cells right next to the future muscles, the myotomes, they also form the tendons.
Ah, so it builds the attachment points too.
It builds a complete structural unit, bone and tendon.
Okay, next up, the outer covering, the skin layer, the dermatome?
The dermatome is made of the epithelial cells that are left behind, you could say. They stay in the dorsal or upper back part of the somite after the muscle precursors migrate away.
And their fate is pretty specific.
Very specific. They migrate just beneath the outer ectoderm, and they form the dermis of the skin of the back and the subcutaneous tissue right under it.
So if you feel a patch of skin on your back, its origin is literally traceable to one of those specific somite segments.
Precisely.
Amazing. And finally, the engine of movement, the myotome.
The muscle-forming region?
Yes.
Precursor cells from the somite migrate just beneath that dermatome layer. And the myotome, well, it ultimately forms all skeletal muscle tissue.
All of it.
Your back, your body wall, and most importantly for complexity, your limbs.
And this is where the segmentation gets even more granular because those myotome cells have to decide something, right?
They do. They have to decide. Am I building an intrinsic back muscle, or am I going to migrate way out into an arm or a leg?
How does that decision get made?
It's all based on a critical geographical line in the embryo called the lateral somitic frontier. It's a border. It separates the somite from the lateral plate mesoderm next to it, and it basically determines if the cells stay put or venture out.
Okay, let's look at the cells that stay home first. The primaxial domain.
The primaxial domain remains medial, so it stays close to the neural tube. It's made up of only somite-derived cells.
And because they stay close to the midline.
They form the intrinsic back muscles, the deep stabilizers, we call them the epaxial muscles, along with the intercostal muscles between the ribs and some neck muscles.
The stay-at-home team, building the deep structure. And how are they wired up?
They're innervated by the most direct route, the dorsal primary rami of the spinal nerves. Simple, organized, close to home.
Now for the explorers. The abaxial domain.
These are the cells that define limb movement. They are the somite cells that migrate across that lateral somitic frontier.
And when they cross that border, they're not alone anymore.
Exactly. They mix and interact with the adjacent parietal layer of lateral plate mesoderm. And this migration is what allows them to create muscles far, far away from the spine.
Which includes basically all of our major movement muscles.
Absolutely. We're talking most limb muscles, the crucial abdominal wall muscles like the rectus abdominis and the obliques, and even structures like the pelvic diaphragm.
And the wiring for these groups, given how far they travel.
They're innervated by the ventral primary rami of the spinal nerves. And this leads us to the single most vital anatomical takeaway from this entire process.
Okay.
Regardless of how far those muscle cells migrate, even if they end up operating the tips of your toes, each myotome retains its innervation from its segment of origin.
Wow. That's an incredible level of organizational persistence.
It is.
So the segment that gave you a sliver of back muscle also sends cells to your hand. And that original nerve root just follows those cells the entire way.
The entire way. The embryonic map is indelible. It preserves that ancient segmented map, which is why even in an adult limb, we can map sensation and movement back to specific cervical, thoracic, or lumbar segments.
But none of this staying or migrating or specializing is random. It's not a choice.
Not at all. It all comes down to a chemical conversation.
Let's get into that. Section 4. The molecular conductors. This is the complex signaling system that controls every decision a somite cell makes.
Right, and this is where we see surrounding structures, the notochord, the neural tube, the epidermis acting like little signal towers. They emit molecules, morphogens, that instruct the somite cells what to become based on how close they are to the signal.
So what signal kicks off the bone-forming process for the sclerotome?
That signal comes from the notochord and the floor plate of the neural tube, which are sitting right below, or ventral to, the somite. They secrete sonic hedgehog, or SHH.
A famous one.
Very famous, along with another molecule called noggin.
So SHH is essentially shouting at the ventral cells, "You are bone now."
That's a great way to put it. SHH induces the ventral part of the somite to become sclerotome. Once those cells get that signal, they activate a transcription factor called PAX1.
And PAX1 is the gene switch.
It's the master switch that kicks off all the downstream bone-forming genes, a very clean chain of command.
Okay, but how does the system handle the muscle and skin portions, the dermal myotome? It has to decide two fates at once, and it's located dorsally, far from that SHH signal.
It is significantly more complicated. It relies heavily on WNT proteins, which are a major family of signaling pathways. The dorsal neural tube secretes WNT.
And WNT acts as the first broad instruction.
Exactly. It activates another transcription factor, PXX3, across the whole dermomyotome layer that marks all those cells for either muscle or skin fate.
But then, WNT needs to be refined. It has to handle two different muscle groups, the primaxial cells that stay and the abaxial cells that migrate. How does one signal do that?
It does it by negotiating with other signals. For the primaxial muscle, the intrinsic back muscle's WNT from the neural tube is the main driver. It directs those dorsomedial somite cells to express the muscle gene MYF5.
So MYF5 is the stay-at-home and build back muscle instruction.
You could say that. But the abaxial muscles, the ones migrating out to the limbs, they need a different genetic program. And they're moving into the territory of the lateral plate mesoderm.
Which is sending its own signals.
Precisely. The abaxial cells need a combined, more nuanced signal. They need WNT, but this time from the epidermis, plus an inhibitory signal called BMP4, which is secreted by the lateral plate mesoderm itself.
So it's an activation signal plus an inhibitory signal.
Yes. This chemical negotiation, WNT activation combined with BMP4 inhibition, is what directs those ventrolateral cells to express a different muscle gene, MYOD.
That is it's highly sophisticated. It's not one switch being flipped. It's a regional chemical negotiation to decide the final identity of that muscle fiber.
It all comes down to concentration gradients and receptor density. A cell interprets the strength of the signals based on its exact position. If you're close to the neural tube floor, you get high SHH, you become sclerotome.
And if you're on the dorsal side?
You get WNT. And the presence or absence of that secondary inhibitor, BMP4, is what decides whether you express MYF5 or MYOD.
Incredible. And finally, what about the skin layer? The dermis. What signals its formation?
That is signaled by a neurotrophin, specifically neurotrophin 3, or NT3. This is secreted by the dorsal region of the neural tube, and it directs that mid-dorsal part of the somite to form the dermal layer.
So we have SHH for bone, WNT and its collaborators for muscle, and NT3 for skin. A perfectly orchestrated three-dimensional command structure.
It's truly amazing.
What an incredible feat of microscopic engineering. We started this deep dive with, 42 to 44 pairs of simple epithelial balls.
Forming like clockwork.
At 3 pairs a day. We watched them differentiate into the sclerotome, dermatome, and myotome, forming the entire segmented skeleton, back skin, and skeletal muscle system. And that whole process was governed by these hyperlocalized molecular signals.
SHH, WNT, NT3. It gives a whole new meaning to the idea that structure dictates function. Every single vertebral ring, every rib, every segment of skin on your back can be traced back to that initial set of segments.
It truly is the ultimate master blueprint.
It is. And if we connect this segmentation to the broader picture of your life, we have to revisit that key rule we talked about.
Innervation.
Myotomes retain their innervation from their original segment no matter how far they migrate. We know this wiring persists into adulthood. I mean, think about the dermatome maps that are used clinically.
Right.
So consider this. If every muscle fiber and every patch of skin you have traces back to a specific geographically mapped segment of the embryo, even the ones that migrated way out into your arms and legs, how do these ancient segmented maps govern pain, sensation, and movement throughout your entire life?
And what does the persistence of that original wiring tell us?
What does it tell us about the foundational, maybe even rigid nature of those very early embryonic decisions?
A microscopic history that defines your entire structure, sensation, and movement, all the way down to your fingertips. Thank you for joining us for this deep dive into somite development.